Good morning again. I told you I have a special surprise, a guest to introduce to you. This is my daughter, Maggie, and I've told a few of you that I've been looking up to her for a while. Stand up. She's been this much taller than me since 6th grade. Anyway, she is a scientist, so I'm having her help me while she's here. She's getting ready to go to Ruth's favorite place. She's going to live there for 5 years. Ruth, you want to tell everybody where that is? Assuming she's here today. And if she's not, Hawaii. She gets to go spend 5 years in Hawaii as part of her schooling to become a doctor. She technically graduated already, but it's a whole bunch of steps you got to go through. Anyway, she's here today to show us how to do a DNA extraction of a strawberry.
Hi everyone. So I'm Maggie, her daughter, as she has just said. I have been into science since high school, so you're actually getting a head start on me. I kind of knew what DNA was, but not really. And so today we're going to learn a little bit about DNA and do an experiment. So the first thing I wanted to talk about should be on this sheet of paper. It's going to tell us all the things that we need in order to do our experiment.
You'll notice it says banana DNA. We switched it to strawberry, but it's still going to work.
Yep. Banana or strawberries, just fine. Whatever you have in the house. Other things you'll need are rubbing alcohol, a Ziploc bag, salt, water that's heated up, and dish soap would be the essentials. Plus probably a glass cup or bowl.
Does it have to be glass or could it be plastic?
You want to be able to see in it, but it could be plastic as long as it's see-through. Okay. So just going down our list here, starting with the things that we know. So all living things have DNA. Even arguably some non-living things have DNA. And all DNA is the instructions by which we're built and created. So just the same as you're going to follow instructions today to do this experiment, our bodies have their own instructions inside of them that helps create everything from your intestines inside your stomach, your lungs that you breathe with, your eyes that you see with. You have to have instructions to put those together, and that's what the DNA is. Okay. Next thing to know is that DNA is inside pretty much every cell of your body. So you have the instructions on how to make absolutely every part of you in every single cell of your body. And it's a lot of information, and in order for all of that information to be kept, the DNA has to get folded in on itself. Demonstrations. Do we have a wire or cord on hand?
Would paper work?
Cord would be better. We have 30 minutes, right? I'm allowed to
Yeah, unless you can stop and go at will.
Oh.
So I found my random cord for this demonstration. So this cord is like a strand of DNA. In humans, it's like one big, long, straight line. However, to fit inside a cell, it's got to be a lot smaller than that. And so there's a couple of tricks that our DNA uses. So the first is that it kind of ravels up on itself like this to make itself smaller. And then that's kind of a mess. So in order to get even smaller, it has to be really well organized. And so there are other things that work with the DNA. And today it's going to be this sucker. So this is called a histone protein. And what it does is it helps the DNA coil up even tighter. And so if we use our histone protein, we can coil up our DNA and make it small enough that it's able to fit inside our cell. So for the experiment today, what we're gonna do is we're gonna try to get this DNA off of the histone protein. Because as long as it's really this tiny and inside the cell, we're not gonna be able to see it. We'd have to be able to stretch it out, make it longer so that we could potentially see it. So we can put this aside.
Ms. Scribner, are they really blue like in our picture?
No.
Darn.
Yeah. It's actually very hard to look at DNA itself. What we're gonna do is just make it easier to see. But if to get close enough where you could see the pattern, like the spiral helix pattern, you have to have a very powerful and special microscope to see it. And there are no colors when you look at something through that microscope.
One thing we've learned about microscopes, which we might not all remember, except I think a few of us probably do, is that some microscopes actually, or some of the processes kill the DNA when they look at it, or the cell when they look at it. But not every one of them does. Are we going to be killing any kind of...
Yes.
Oh.
We will be destroying the cell. But the good news is when you already have a strawberry, the plant doesn't really matter. Like, the plant doesn't care. The strawberry is not going to be upset that it's losing its cells. That's what it wants, actually. So it'll be okay. Alright, so anytime we're doing an experiment, we want to have a question in mind, and we want to consider what the answer to that question might be. In science, we call this question a hypothesis. And we consider our answer to be like an educated guess about what's going to happen. So the question we have on this sheet is, can we see DNA without a microscope? Do you think we can see DNA without a microscope?
Well, I don't think so, because the only place I've ever seen that pattern and been told it was DNA was on paper. So if what I'm looking at isn't really DNA, then I think I probably can't see it.
All right. So you've had an answer to the question supported by things that you already knew, and now we're going to do the experiment to see if that is true or not. Alright, so step one here is put the rubbing alcohol in the freezer. We've already done this. It's not 100% necessary, but it does work better if the rubbing alcohol is cold. So step number 2, Mademoiselle, can you select a few strawberries?
More than one?
Yes. Maybe 2 or 3. Put them in our Ziploc bag.
Does it matter if I go with one that's really still fresh looking compared to one that looks like I should have eaten it a few days ago?
Probably not.
Okay. I'm going to go with the one that looks like I should have already eaten it. That way I'm not losing a strawberry I really wanted. Is that enough? Or
That's fine.
okay.
Alright, so the next is the fun step.
Even without the alcohol?
Gotta smash them.
Okay. Can I step on it to smash it?
Mm-hmm.
It doesn't matter? Okay. Ew. That feels wet and cold. Is that good enough?
No.
I was afraid the bag would open.
And while that's happening, I'm going to start warming up our water.
So the water needs to be warm, but the alcohol needs to be in the freezer.
Yep. Hey, Did you know that we have a filmographer who's helping us today?
Mr. Scribner. He's behind the camera.
Hi.
This will be much faster.
So, something that Ms. Scribner, the scientist, may not know is that Arthur knows Mr. Scribner personally. I assume Arthur's here today watching this because he's almost always been in class since we've had to go to these video online class settings, but
I got it, don't worry guys.
Mr. Scribner used to take last school year, used to take or go with Arthur and Juan and Sebastian. No, Sebastian didn't get to go, did he? Anyway, to go play bowling. What you looking for?
Thing that I got out and forgot where I put it. Okay.
Is this good enough?
Nope.
You're kidding.
Nope. Alright, I'm gonna make it a little longer.
Maybe that's why they said bananas. What you looking for?
Just remember you used to have a rolling pin.
Oh,
This is fun. Yes?
so, excuse me, Mr. Scribner, I'm gonna get alcohol.
So now that I have hot water, I'm going to mix in quite a bit of salt. Goodly amount here. Did you get me por favor?
By the way, you all are officially on your first online virtual field trip at my house. Glad you could make it.
It's a field trip. Then have you introduced them to the doggy and the kitty cat?
Well, not yet. We're more interested in this, but we'll get to that.
Oh, I'm interested in doggies and kitty cats. Okay, so there's enough salt in it that it's no longer see-through. You can see the whiteness of the salt. You want a lot of salt. Why is it skipping steps?
Double-sided page, maybe? Step 3. Step 4. Step 1.
Yeah, it kind of skips the dish soap. Dish soap would come first, yeah? That's what I remember. Okay. It's a good thing you know this. Scientific folks.
And why are you doing this so?
Okay, so there are two things that we need to get rid of in order to get down to the DNA. The first being the actual cell itself. And the cells that our bodies are made up of are kind of fatty. Like when you eat meat or you eat potato chips, how there's still grease left on your hands. And we use dish soap or hand soap, but dish soap's better for it, to clean those things off. And so that's what we're doing here. We're gonna clean off the cell by using the dish soap.
That is a lot of dish soap.
Yeah. It's foamy already. You wanna mix it up?
not quite closed.
That looks pretty cool.
It's kind of pink now.
I don't understand.
Mostly what he says I don't understand.
All right. So now we have our dish soap all mixed up. We can add our warm salt water and hopefully not spill it all over ourselves.
Oh, I think there's a hole in the bag.
No, then let's do it over this.
Yeah, that is hot water.
Indeed. Okay, so I said there were two components. The second one is the histone proteins that we demonstrated with our little thingamabob.
Oh, that was a histone protein? What is a histone?
It helps hold the DNA coiled up really tight so it can fit in a cell.
Oh yeah, you already said that.
Can you just talk like a normal
I mean,
person?
yeah, because like it's not helping. We don't know what you're saying.
All I'm seeing is the back of your head.
Well, because it's hot water. I'm holding it as hard as I
That's
can.
Switch over to the other side where Mom was standing. There you go. Now I can see.
I know. They're just losing their mind.
You always have to copy your cat because, you know, copycats. Okay. Very sure you can put it back in here. I like that one. Okay. Do we have a coffee filter? Or strainer?
I mean, it's got coffee in it.
Do we have a strainer?
On the wall. The cat. Hey, Gertie. Hello. Say hi to everybody.
Maker.
And that's Luna. Actually, she did.
Oh.
That's our puppy dog.
Okay. So now I'm going to take this big goopy mess and put it either through your coffee filter or your strainer.
Gertie, what are you doing?
Whatever she wants to do.
She's sniffing my feet. Meow to you too. Gertie. Hello. You were very photogenic. You know that?
Skye ate her dinner and she's a little bit upset about it.
Where was she?
I have no idea. But she will not leave Mom alone. I mean, Miss Scribner. Alright.
By the way, we're right at 20 minutes.
Okay. Mr. Cameraman,
Mm-hmm.
I can put in my alcohol at this point, yeah?
Okay.
Right?
Mhm.
That's what you recall?
That's what I remember. And then it precipitates out.
That's what I remember.
Okay.
What does precipitate mean, Mr. Cameraman?
Isn't it before the precipitate?
In this case, it means it's becoming a solid and condenses. And you get it,
Condenses.
and you get to see it
What does that mean, Mr. Cameraman?
that they come together.
Why didn't you just say that?
And what is they?
The DNA.
That's a lot of alcohol.
Yeah, because we want it to be pretty cool. Alright. Come hither, cameraman. Alright, so do you see all this white stuff that's floating to the top?
Hold on. It's really grainy. No way. That's all DNA?
Mm-hmm. And there should be... Okay. I don't know if you're able to zoom in close enough, but you can see how it's kind of stringy.
Mm-hmm.
Hold still. Yes, I can.
Yeah.
It is stringy.
So sometimes when you're really good at this, you can actually get like super big strings, long strings, that kind of hang down into the water when you pick it up.
If you get a little bit longer.
Or if we get it a little bit colder, that might help too.
Toothpicks work really well.
You're totally allowed to talk.
They usually cling to the toothpicks.
Yeah. But it's still too warm. Trip already and something else. This looks like it would taste delicious.
Probably salty
Cool.
and rubbing
Hold on.
alcohol.
Hold it back up to the light. Hold it up here.
Oh, wow.
Can you see it swirling around in there?
Yeah.
So not all of that is DNA, but some of it is.
The white stringy stuff is DNA.
So I was wrong. You can see it without a microscope.
Yeah. In the right conditions. I'm getting a little bit, but it's not very satisfying. You can let that hang out for a bit, I guess. In the meantime, see what the rest of these questions were.
That is so cool, though. It's a good photo.
All right. So like you said, okay, first of all, did you say if you could see the DNA with a microscope or not?
My guess was that we could not see it without a microscope.
And were you correct?
I was wrong.
You were wrong. So does that mean that the experiment was bad since you were wrong about what would happen?
Well, no, I learned something.
Right, yeah. Kind of, if the experiment, just because it didn't happen the way that you thought it would, doesn't mean that it was a bad experiment. Either way, you learned something new about the world that you didn't know beforehand. So some interesting things about what we've been talking about is that the microscope was developed by Robert Hooke in 1665, and that's whenever we started to discover that cells were what made up living creatures, because it's not just humans that have cells. The strawberries had cells. That's where it kept its DNA. Luna Bear, over there on the couch. Luna Bear has cells. Lots of people have cells. And so then as we get closer and closer to now, we get more powerful microscopes and we're able to see smaller, more detailed things. So DNA is really, really small, really, really detailed. So usually, in order to look at the structure, the spiral structure, you have to have a very, very powerful microscope. That's quite expensive.
Heredity.
The concept.
Mm-hmm. Yeah.
All right. Can I just go over this?
Sure.
We're just okay. So this is a worksheet, a quiz on genes and heredity, and
you will be taking that quiz, so pay attention.
so for question number 1, it says, the passage of traits from parents to offspring is called. The answer to this question is heredity. And heredity is why kids look like their parents or act like their parents. Because their parents had genes that were on the DNA that got passed down to the kids. And so their kids are a combination of both parents. And that's called heredity. Question 2 is, the traits of an organism are passed to its offspring on small coded DNA strands of material called, and this is genes. So genes are the specific traits. An example of a gene might be if you have brown eyes, you have a gene for brown eyes. And probably, definitely, at least one of your parents has brown eyes. In order for you to get the brown eye trait, one of your parents had to have a brown eye trait. And that trait is on DNA called a gene. Number 3, Watson and Crick are the scientists who discovered. See, he did it too. He did it too.
Oh, well, who is not Watson and Crick?
Watson and Crick are credited with the discovery of the structure of DNA. However, they were using data from an experiment conducted by Rosalind Franklin.
Ruth, did you hear that? A girl scientist.
She was a pretty awesome scientist. She was working in a lab in which there weren't a whole lot of other women. And she was in charge of her lab, but Watson and Crick thought that they were supposed to be in charge of her lab, and so they took her data that she collected without asking for it. And then they published that information, and now they're famous, even though she's the one who took the pictures of DNA to begin with.
Well, you don't have to know that for the quiz. You just need to know that because it's good knowledge.
So the answer to this question is that Watson and Crick discovered the double helix structure of DNA.
Huh.
Question number 4 is, during sexual reproduction, each parent contributes. And the answer to this one is 1 strand of chromosomes or half of the new organism's DNA. So like I talked about earlier, I look a lot like my parents because I am half of my mom and half of my dad, who I happen to actually look. Oh, hold on, family photo. Hey.
So Mr. Scribner and I both have blue eyes.
And I.
So does she.
So actually, I think those are probably all the important ones for this experiment. The others are kind of necessary.
29 minutes, 4 seconds.
29. So like perfect.
Awesome. Okay, well, thank you for helping us understand all that, Ms. Scribner.
You're welcome.
And Mr. Scribner, thank you for holding the video.